All right, welcome everyone. Glad you could be here. So we just have a couple more slides left in the vitamins lecture, and then we'll go on to minerals. So hopefully you noticed that study guide's not due till Wednesday for this one. So I understand the last part here, we won't have a review for those questions. So if you have questions on that last section we'll go over today, that anything that's unclear, send me an email or something and I can help you through the question. So we ended on thiamine, and there's not a lot that is interesting about thiamine from a food science perspective, except that it's super unstable and it's more stable in acid. But degradation is pretty significant usually, and especially when it's heated. So typically what happens is that methylene bridge between those two rings, that is what's cleaved, and then you end up with these two rings that have no vitamin activity anymore. So on to vitamin B2, which is riboflavin. So riboflavin takes on all of these forms. So these would all be active forms of riboflavin. The most common one would be flavoquinone, the oxidized form. This is the one that we will typically see. And when, I don't know, in lab, sometimes we measure riboflavin in lab, and we measure it in milk or some kind of dairy product. So dairy is actually really high in riboflavin. And if you, I don't know, sometimes with yogurt or something, you'll see the watery layer on the top, the whey separating out. So if you were to maybe pour that off, you would see that it's slightly yellow. And that is because of the riboflavin. And you could actually see if you took that yogurt, if you took milk out in the sun for about an hour and then brought it back in and you were to, you could separate out the whey from the yogurt or precipitate out the proteins from the milk, you would see that it's no longer yellow because that riboflavin has degraded. So the key with riboflavin, it's pretty stable in the dark. But as soon as it's exposed to UV light, it is very unstable. So, that's why we have milk containers that are, frosted plastic, not transparent plastic. And that helps to protect that milk from light. Of course, better is the paper carton, which is completely opaque. And so that will preserve it even better. So really the strategy to preserve riboflavin is simply to keep it in the dark. So if you have an opaque packaging, that will help to keep it from degrading. So as I mentioned, there's some forms of the vitamin that start with the prefix lume for light. And so those derivatives don't have vitamin activity anymore. So if you see lumeflavin or lumichrome, those are degradation products of riboflavin. So they no longer have vitamin activity. Okay, niacin. So these are the two forms of niacin here. They comes in both forms. The niacinamide, that would be the active form, and also the form that's most common. So this is This is a little challenging having two presentations up here. So niacin is that vitamin that I mentioned that, you know, when we talked about pellagra in the United States, people eating corn ended up in mental institutions for just having a niacin deficiency. One of the symptoms is dementia. So, you know, corn there, we see very, very low and free niacin. But if we just boil that product, or better yet, if we treat it with lime, so this is like the traditional Native American way of producing or consuming corn, would be to soak it in lime, calcium hydroxide, it actually, yeah, will break the linkage. So corn, the niacin in corn is present in a complexes relatively not well known, but probably complexed with polysaccharides and proteins. And it's probably ester linkages because they're very labile to alkaline conditions. So soaking that corn in calcium hydroxide will free the niacin, and then, you know, you wash off the calcium hydroxide, grind it into a dough, and that's your masa flour that you, or your masa dough that you use to make corn tortillas and stuff like that. So yeah, you see that corn is a really good source of niacin, but if we just eat it straight or even boiled, it's not really that good of a source. So treating it with lime really frees that niacin and enables us to get that vitamin. Have their coffee, you know, the darker the roast, the more niacin there is. So niacin is stable to pH changes. It's stable to heat treatments. It's just bound in a lot of things. In coffee, it's not bound to the proteins and the carbohydrates. It's a methylated version. So here you've got like a methyl ester there on the acid group on nicotinic acid. So if I point here, you've just got like a methyl group here, methoxy group. So roasting it will break that bond and free the niacin. So that becomes a source of niacin. Vitamin B6 is a vitamin that is rarely something that people are deficient in. Typically, we get plenty of vitamin B6 in our diet. So pyridoxal would be the active form of the vitamin. But typically in foods, we're going to see more pyridoxamine. And the alcohol version's a little strange. I would probably call it pyridoxal, but it's called pyridoxine for some reason. But though the amine form is the one that we see really commonly in foods. Heat-treated foods. So anything that we cook or boil will have the amine form of vitamin B6. And so actually vitamin B6 is interesting from a food science perspective, not because it degrades or because it is you know, something that people don't get enough of. It's because it reacts with the amine containing compounds in food. And so what are those? Typically proteins or free amino acids. So pyridoxal, so the aldehyde version, will react with an amine group, any amine, but in foods we have lots of amino acids and amine groups on proteins that are floating around. And so when we heat a food containing vitamin B6 and protein or amino acids, we get a reaction called transamination. And I put a box up here to write that down, but it's not showing up here. So I don't know if those of you online are having a hard time hearing me or spelling it, but transamination, just the process of transferring the amine from one compound to another compound. And then in the place of where that amine was, we get a ketone group. So here, the aldehyde and the amine react. And I should point on the screen. So these two react, just as shown here. And eventually, what we get is an amine on the pyridoxal. So now we have pyridoxamine. So that's why I say in foods, we have mostly this. And the amine came from some amine-containing compound in the food, either a free amino acid or could have come from lysine on a protein. And we have this derivative of the amino acid. This is called an alpha keto acid because it has a ketone group here on the alpha carbon from the acid. So it's an alpha keto acid. You do want to be able to recognize these alpha keto acids because they'll come up over and over again. Actually, this transamination reaction comes up over and over and over again. So, you know, all the intermediates, you don't need to worry so much about those. You just want to know what the products of the reaction are when you react these things together. So the amine basically moves over here where the aldehyde was, and then we get a ketone in its place. And so this is important from a nutritional perspective because vitamin B6 can decrease the biological activity of proteins. Because basically in a protein, we don't have a lot of free amines. We have, you know, the N-terminal free amine, and then we have all the other amino acids have their amine tied up in an amide bond between another amino acid. So pretty much we have one amino acid that does have another free amine, lysine. And lysine is an essential amino acid. So if we do this reaction and the amine comes off of lysine and goes on to pyridoxal to form pyridoxamine, then we've lost some of the biological activity, some of the nutritional quality of that protein because it no longer has lysine. So also pyridoxal can react with sulfur-containing amino acids. And it does that in this manner. So the reaction is the same as we saw before. So all these intermediates, you can see these intermediates are the same as those that we saw up here. But if we have a sulfur-containing amino acid, and this is just a sulfur-containing amino acid, it's not a sulfur-containing amino acid in a protein. It would have to be free, in this case, cysteine. So this amino acid here is cysteine, free amino acid. And when it reacts, this amine on the cysteine molecule will form pyridoxamine, just like we saw here. So this compound is formed. But in the process, the sulfhydryl group comes off of the cysteine residue. And we generate hydrogen sulfide. And hydrogen sulfide is a strong smelling compound. It's a very undesirable smelling compound. But in trace amounts, this is going to be really important to the flavor and aroma of cooked meats. So vitamin B6 is playing a role in generating the compounds we're familiar with when we consume or when we smell cooked meats. So through this reaction, you want to understand that pyridoxamine is formed, and it still has vitamin B6 activity. So we haven't lost any vitamin B6 activity. But we can lose, protein quality. So, cysteine is an essential amino acid. So is lysine. So when that reaction occurs, the protein quality goes down. But we still have vitamin B6 activity. Okay, next we have folates. Folates are highly unstable, another unstable vitamin. And it's actually pretty susceptible to acid, but it does prefer to have pH slightly acidic. So something between about like 5 would be optimal for this. 4 to 7, 4 to 6. Slightly acidic. If you get super acid or anything above 7, it's not going to be stable. Very susceptible to heat. The table down there shows several different functional groups that can be added to either carbon 5 that I've numbered there or 10. And in some cases, they can even form a ring. But those would all be active forms of the vitamin. And they're listed in order of stability. So the top one is the most stable, the bottom one is the least stable. That bond between what I've labeled as carbon 9 and 10, that would be where the molecule breaks down. So similar to thiamine that has these 2 rings attached together, this one also, the susceptible region is that bond in between the 2 rings. I guess I can mention the top one, folic acid, that would be the synthetic form of folic acid, or the vitamin. And then the bottom one is the naturally occurring form. And so it's a little bit strange because folic acid is the synthetic form, whether or not it's protonated. So it's not like it's folic acid when, when this is protonated, it's folic acid, and when it's ionized, it's folate. It's not like that. So if you have the synthetic version, it's folic acid. If you have the natural version, it's folate. And we just have this chain that comes on here. This is glutamine. It's polyglutamine stuck on there if it's the natural form. All right, biotin. So I have this protein here, avidin. And avidin is a protein that binds very strongly with biotin. And so if we consume this protein in its native state, it will bind biotin and we can become deficient in biotin. Almost never do you become deficient in biotin. It's like vitamin B6 or, like we're going to talk about in a minute, pantothenic acid. These vitamins we're almost never deficient in. But if you consume a lot of this protein without cooking it, then you can become you can become biotin deficient. Anyone know where this protein comes from? So this protein is present in egg whites. So consuming raw egg whites can lead to a biotin deficiency. If we cook the egg whites, the avidin denatures, it doesn't bind biotin anymore. This is actually the basis for many reactions that are done in the lab to measure certain things. So you can attach some protein, or you can attach some molecule to biotin and then add avitin, and it can pull that out. So you can isolate things and stuff like that. It's very common in biochemistry. Okay, pantothenic acid. This is one that we don't need to worry too much about. This is a ubiquitous vitamin. That panto prefix means everywhere. So it is a ubiquitous vitamin. It's a component of coenzyme A. So if you're familiar from biochemistry with metabolism, like acetyl-CoA or something in the TCA cycle, this is this portion right here. This is a portion of coenzyme A. So important from that perspective, but it's stable in food processing conditions, and it's present in many, many foods. So we don't really need to worry too much about pantothenic acid. Okay, finally we get to vitamin B12. So this is a very complex vitamin from a structure perspective, you can see. So, but what you immediately can notice is that it has that trace element in the middle here, cobalt. And that cobalt is coordinated inside of this ring structure. And this coordinate here is called a chelate. Actually, we're going to talk about them in detail in just a minute when we get to the metals. But it's called a chelate. So it starts with a CH, but you should say it like it just starts with a C or a K. So chelate. And so that's when a metal ion is coordinated with a ligand in several places. And you see that here. So this cobalt is coordinated with all of these nitrogens, as well as this R group, and then this nitrogen down here. So 6 places where it is bound to this molecule. And actually, 6 locations of interaction is the most common with these chelates. So we have several different things that can attach to that cobalt at the R position. And so that's what's listed there. So the synthetic form would be cyanocobalamin, where cyanide is coordinated with that cobalt. Cyanide is definitely toxic, right? But in vitamin B12, even if we consume 1,000 times more vitamin B12 than we need, we would get less cyanide than we just get from regular food. So it's not going to give us a lot of cyanide. In fact, if someone becomes or gets a high dose for some reason of cyanide, the treatment is to give them one of these other forms of vitamin B12. And then once that gets inside of us, it's going to complex with the cyanide, and then you pee it out. So it can actually save you from a cyanide poisoning. In the body, what we want is the methylcobalamin. So the job of vitamin B12 is to move methyl groups around. So the active form would be the methylcobalamin. But typically, if we're going to add it, it's going to be the cyanocobalamin. This vitamin is only present in animal foods. So meats and milk and eggs and stuff like that. So this is a vitamin that people that are vegan are always really concerned about getting, because if you completely eliminate animal food from your diet, this is one that you cannot get. So you have to take it as a supplement. Vitamin A is also only present in animal foods, same with vitamin D, but you can get those either from carotenoids in plants or from sunlight. So you don't have to worry about those. This one, there's no other way to get it but from animal foods. All right. Any questions on vitamins? So remember, if you have any problems on the study guide, since we're not going to have a review on those last few vitamins, if you have any questions, please feel free to email me and I will help you through those questions that you might have. Okay, let's talk about minerals. Minerals I always get a little bit uncomfortable with because when we're talking about minerals in foods, we're really talking about elements. So I have this hard time with the terminology that we use. But you know, those things in purple and then maybe some of them in green, those are elements, right? But when we say minerals, we're talking about iron, magnesium, sodium, which are elements, not minerals. So just to be clear, when we're talking about these elements, you know, typically we'll refer to them as minerals, even though they're technically not minerals. So let's see. Most elements in foods are present in some sort of complex. So they don't occur by themselves. So, and actually I should say, when we talk about minerals in food, we're talking about all these things except for the carbon, nitrogen, and oxygen. So these three would not count as minerals in food. They're present in all these organic molecules. So we're talking about everything except carbon, nitrogen, and oxygen, and hydrogen. So most of these minerals are going to occur not free, but bound to something or complexed with something. And so, except for a few, so sodium and chloride are going to be free. So they're actually found in some foods, but a lot of times they're just added. And whether they're added or naturally occurring, a lot of times they're free ions. Sodium, chloride, and potassium. And then they're also present in chelates, which we just saw for cobalt. We'll see that for some of these others in just a minute. They're also present as complexes, which is like a level below a chelate. So a chelate would be if you are coordinated in several places with that element, and a complex would be if you're just coordinated in one place. And then they're also present in anions, so like phosphate, There's a couple slides, we'll see that. But like multi-element anions that are typically oxygenated. So phosphate and sulfate and stuff like that. Okay, so this is the recommendations and its daily value. Oh, you know what? I should have turned off that light. You can't see very well. So this is daily value. And just like for the vitamins, we have the old one prior to 2019 and then the new one. And just like for the vitamins, we see some of those recommendations went up. So like phosphorus, I think potassium, potassium didn't have one before. And then calcium. Some of these went up. And others went down. So as we get more information, we realize maybe we don't need as much as we thought. Here's some sources of these different nutrients. So always just a good idea to know kind of where some of these come from. Dry beans and whole grains, those seeds that we eat, are really, really high in a lot of these minerals. So magnesium, zinc, iron, molybdenum, magnesium. So a lot of these are really, really high potassium in these seeds that we eat. Of course, you know, calcium is high in dairy products, but we do want to think about leafy green vegetables as a source of calcium. So there are other places we can get it. It's a lot more difficult, but we can. Some of the underconsumed nutrients are minerals. So calcium, magnesium, potassium, and iron would be all nutrients that we're concerned about that are minerals. So here is how these elements occur in foods. Besides the sodium, so the sodium here is shown as a complex, so it can occur this way, but it comes off and goes back on freely. So a complex here is what I mentioned. It's below a chelate where there's just one interaction between a molecule and the ion. And then a chelate is when we have more than one interaction between the element and some molecule. So in this case, we have two interactions. And then they are present as oxygen-containing anions like the phosphate there. So I do want to just say, if we have a metal ion that is complexed or chelated with something, then that other thing is called a ligand. So in the first case there with the complex, I gotta stop pointing at the screen and point here. So the first one with the complex here, the acetate group is the ligand. And here this molecule is the ligand. This molecule happens to be oxalic acid, or in this case, oxalate. And oxalate is really important. It's present in all those leafy green vegetables. And so it complexes with calcium. And so unfortunately, those leafy greens do have a lot of calcium, but it's tied up in this oxalate chelate. And this oxalate chelate is not soluble, so they're very difficult to absorb. Or as anions. So remember that term ligand, it's just the other thing that's not the metal. I don't know how to go to the next slide here. Okay, so here we have a picture of calcium EDTA. Stands for ethylene diamine tetraacetic acid. It's a kind of a long name. Typically, we just see EDTA. So this is a very common chelate, a chelating agent to add to foods. So you'll see it everywhere. All kinds of sauces, anything, especially stuff that has lots of water, like sauces or meals, soups, stuff like that, often will have EDTA in them. So unlike the oxalate chelate that we saw here, EDTA chelates are soluble. And so if you have a metal like calcium here that is complexed with EDTA, it doesn't inhibit absorption. We can still absorb it. All right, so let's go through some of these. So metal cations are electron pair acceptors. They're positively charged, so they need some negative charge. So they're going to look for electrons. So anything that's going to have a negative charge or a partial negative charge is going to attract these metal cations. So in most of these organic molecules, proteins, carbohydrates, plant secondary metabolites like antioxidants, these metals are typically going to bind to oxygen or nitrogen because those two elements in these biological molecules have partially negative charges. And so you see here in EDTA, complexed with nitrogen and oxygen. And typically in 6 places, like it has in that last bullet. So there's a couple of rules that I'm going to give you right now that will determine the strength of that chelate. Okay, so one of them would be the ring size. So you can't see it great here, but maybe in some places you can. Do you see this ring right here that's formed with the calcium and the oxygen, nitrogen? 5-membered ring. If a ring is formed that's 5 or 6 members, that's a good thing. So increased stability. So if we have a ring size of 5 or 6 members, that's going to increase the stability of that chelate. So we saw in that oxalic acid, if you look back, I'm not going to turn back because I don't want to lose this, but there's a 5-membered ring there, so that would be very stable. Okay, next, number of rings. So we see here in this EDTA, we see this 5-membered ring. There's another 5-membered ring right here. There's another 5-membered ring down here. Another 5-membered ring. So this one is super stable, right? So the more rings, not nor. More rings, more stable. Okay, number 3, charge. So if we have a formal negative charge on any of these interactions, that's going to be stronger than if we have a partial negative charge. So in this case, we have all, well, this would be a negative charge here. So we do have some formal charges. So charge, okay. Next would be just properties of the donor atom. So, for instance, I mean, this is kind of along the lines of 3, but oxygen is more electronegative than nitrogen. So the more oxygens we have, the more stable or the stronger the chelate is. I'll just write electronegative. Okay, so resonance. If the electrons can be distributed in many places around that ring, so if we've got some double bond characteristic, for instance, then that will be a stronger ligand. So resonance and ring, stronger. And then 6 would be size of the whole thing. So this EDTA is a small molecule, say compared to a protein or something like that. So you can imagine that a protein could have several partial or full negative charges on it. And so it could bind in several places with an ion and form a chelate. But if there's a big area there, then it can wobble around and fall apart. So if something is smaller, it's kind of in that fixed position, it will be more stable. So the size, small, more stable than large. Okay, so, and that's really just because things can line up and kind of lock in that position better than something that can wobble around a lot and fall apart. Of course, you can reach a limit, right? If it's too small, then you may have issues with steric hindrance, like maybe 4 or even smaller rings. Of course, that would not be favorable. So here's an example. This salad dressing here has our EDTA in it, somewhere in here. Here it is, disodium EDTA. And what does it say is the role of the EDTA? It's a preservative. What time is it? 48? Okay, we'll end on this slide. Okay, so it says it's preservative. How is it a preservative? Does it bind to a bunch of pro-oxidants? Yes, good job. Binds to pro-oxidants. So I've mentioned this in class already. What are those pro-oxidants? Metal cations. Okay, so when we have free ions floating around, they can be pro-oxidants. And so the EDTA is going to prevent that, so it's going to be a preservative. So not a preservative from a microbiological perspective, but it's a preservative from a chemical perspective. So anyway, whenever we have any kind of ions in food, they need to be chelated to something. So they need to be chelated so they don't catalyze lipid oxidation, or actually free iron. You know, iron is something that is often limiting. You know, it's limiting for us, where often we don't have enough iron. It's also limiting for bacteria. So if we have lots of free iron floating around in a food, bacteria can grow more easily or readily than they normally would be able to. So we need to chelate that iron so that we don't get uncontrolled bacterial growth as well. I'll just finish off here a couple of things. So besides EDTA, you know, we have oxalic acid that we saw a picture of. We'll see one again tomorrow. There's other chelates, other organic acids that form chelates. Chelates also, you know, in our vitamins, like the cobalt. So also very common in proteins as a coenzyme. So a lot of these elements will bind to enzymes to either activate those enzymes or deactivate those enzymes. So that would also be another chelate between those proteins and those ions. They're also present in pigments. So myoglobin, we have an iron there. Chlorophyll, we have magnesium in chlorophyll. We'll see that when we talk about color in a couple of weeks. So they're always chelated to these things. Okay, thanks everyone. That's all, unless anyone has quick questions. All right, good. Well, we'll continue. We'll finish up minerals hopefully on Wednesday. Mm-hmm.